This is TWIS. This Week in Science, episode number 842, recorded on Wednesday, September 15th, 2021. Science for Inspiration. Hey everyone, I'm Dr. Kiki, and tonight on the show, we will fill your head with cannibalistic butterflies, a mammoth solution, and help for aging. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. The following program contains controversial subjects such as facts supported by science. If you are one of those people who somehow feel threatened by facts supported by science, know that you are not alone. Many people in the past have had irrational fears of the known. The Earth was known to be round since before the days of ancient Greece. A Greek astronomer, who would be a household name if not for having a name that is nearly impronounceable, estimated the Earth's circumference to within 15% in the year 240 BC. His major flaw? Assuming the Earth was a perfect sphere. We're bulgy round in the middle, and we like it that way. But if it wasn't for that, he'd have been exactly right. Ancient sailors before this time likely understood that the Earth was round, as ships at a distance have a habit of sinking into the horizon. Yet, some people, more than 2,000 years later, still have trouble accepting that the Earth is round. Despite Giordano Bruno and Galileo Galilei, both attesting to the fact that the Earth orbits around the Sun and not the other way around, many still who wore robes and to work were feeling less special because of it and wanted them imprisoned, murdered, and forced at least one of them to recant. So, being afraid of facts supported by science has a long history. The question is, what do you do about it? The best thing to do, in the face of the fear of the known, is to simply learn more about it yourself. Take some basic science classes, watch a few science documentaries on a subject, or read books written by scientists involved in the work you are fearful of. If learning isn't your thing, that's okay, I get it. But then I have a question. When did this happen? You can no doubt tie your shoes, maybe drive a car, possibly even hold down a job. So learning must creep into your life on occasion. And if you try, with science, you might just find something interesting and that learning isn't so bad after all. And if there's one thing we know for sure that we don't ever need to fear, it's that This Week in Science is coming up next.
I've got the kind of mind that can't get enough. I wanna learn everything. I wanna fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I wanna know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again to talk about all the people that are up in space right now.
Oh gosh.
There
I
are
watched
many.
one of them one or two of them walking around out there doing like a repair thing this week.
Right. There are not only people out in space, there are people in the International Space Station. There are people who were launched up by SpaceX. Yes, SpaceX's Inspiration 4 today launched its first civilian crew up into space. This is all civilian, totally space tourists. Do we call them amateur astronauts? Do we call them astronauts? Do we call them space tourists? I don't know, but there were 14 people in orbit around the planet today. I mean, if this isn't a day for people in space. Right. But they're up there. We have escaped the gravity well of our planet. We're putting people into space, and it's not just highly trained astronauts. Amateurs, people who want to go there, can pay for it if they have the cashola and can get themselves into space. So what does this mean for the future of humanity? I don't know, but today isn't a day for Inspiration 4. and science. So we have a lot of stories today. I have stories about fixing aging. We want to fix aging. Blair, I have like 3 stories that are just for you
I'm gonna take
on how
notes.
we're going to make your old age a better old age. Okay. I've also got an influence of black holes and a rundown of the Ig Nobel Prize winners.
That's always fun.
So
No
much
way, it's
fun.
already time for that. That's been a year?
I know, it came back around again, and I'm like, wow, how did that
My
happen? But-
Lord. Okay.
yeah. All right, Justin, what'd you bring?
I've got ancient seas on Mars, a mammoth solution to global warming, just good news, COVID edition, and why your cells are so sticky.
I have sticky cells.
Yeah.
Oh, I didn't know that.
Well, have you ever had a cut? Did you just ooze out?
Oh, okay.
Mm.
That's right. There's something. It's not just
Something.
surface tension that keeps all the
No.
stuff in. Okay, Blair, what is in
Yes.
the animal corner?
Yes. I have toilet trained cows. I have cannibalistic butterflies. And then before that, I have a fun story about humans and heartbeats. Just for fun.
Oh,
I always want to stop the show
Love
Laura,
them.
and like
Love them.
hear at least one of your stories each week. Just like okay, let's just do that one right now.
To the
I can't wait.
chase.
we can't
Maybe I'll
wait
have to
until
wait.
later, but we
Have to wait.
can. We can wait until
It's the
Blair's animal corner.
marshmallow for you,
Yeah,
Justin.
that's
Yeah,
right.
it is. It
No,
is.
hold
It's
on.
every time. It's right away.
As we jump into it, I do want to remind you, jumping in, that you can subscribe to This Week in Science everywhere that we are found on podcast platforms, on YouTube, on Facebook, on Twitch, and on Instagram and Twitter. And there's so many places. You can really find all the information at TWIS.org. Are we ready for the science?
Yes.
Yeah.
Yeah, all right, diving into the big pool of science. I'm going to start us off with the Ig Nobel Awards. There's nothing ignoble about them. They are fantastic and fun, and that's the whole idea of the Ig Nobel Prizes, is that it's a celebration of the weird and the unusual that makes people laugh and hopefully think a little bit. This year's prize winners, they were awarded last Thursday online, and the biology prize went to Sweden for a paper entitled A Comparative Acoustic Analysis of Purring in 4 Cats, a phonetic pilot study of vocalizations in 3 cats, a phonetic pilot study of chirp, chatter, tweet, and tweedle in 3 domestic cats, a study of human perception of intonation in domestic cat meows, melody in human cat communications, meowsic, origins, past, present, and future. They, of course, won the Ig Nobel Prize for Biology for analyzing variations in purring, chirping, chattering, trilling, tweedling, murmuring, meowing, moaning, squeaking, hissing, yowling, howling, growling, and other modes of human-cat communication. So fun, kitty cats. The Ecology Prize went to Spain and Iran for using genetic analysis to identify the different species of bacteria that reside in wads of chewing gum that have been thrown away and gotten stuck on pavements. Yeah. What kind of bacteria?
That sounds like a really good study. I don't know what's ignoble about it. That sounds like, yeah.
What kind of bugs? Yeah, what microbes, what bugs are in the gum that you chew and that you leave on the pavement and that potentially get smashed by people walking by?
That or
brought
like,
into
yeah,
their home on their shoe.
Yeah. And their paper was called The Wasted Chewing Gum Bacteriome. I mean, that in itself is the title of the next best-selling, New York Times best-selling novel, I think. Chemistry, Germany, UK, New Zealand, Greece, Cyprus, and Austria shared this prize for chemically analyzing the air inside movie theaters. You want to know why?
Smells like popcorn.
Not just to analyze popcorn and how much popcorn people are eating. Oh, no. To test whether or not the odors that are in the air indicate the levels of arousal from violence, sex, antisocial behavior, drug use, or bad language in the movies that people are watching.
Oh, they were looking for pheromones.
They were looking for pheromones or
That's
any
great.
kind of odorant that would be, I mean, yeah, we sweat more, we get excited, or we're, who knows? There's a lot there, human behavior. The KidneyX Prize, Japan, Switzerland, and Italy shared for mutual anticipation can contribute to self-organization in human crowds, which really was to figure out why pedestrians run into other pedestrians.
Oh,
What happens? What happens other than staring at your iPhone or whatever it is to make you run into somebody else when you're walking?
That's interesting. I don't know that's got to be a kind of rare event. Or I guess I live somewhere with not enough people.
I was going to say something controversial and say it's men who expect women to get out of their way.
Ooh. Well, another thing that I have
women
learned
also don't get out of the way. See, like it's both ways when people run into each other.
I have learned that if you hold eye contact, you will probably run into the other person because you predict that they're going to go a particular direction and they predict you are going to go a particular direction. And you end up kind of correcting based on your prediction and walking into each other.
That's why every New Yorker tells you not to make eye contact
don't
with
make
the driver
eye contact
when you enter the street. Otherwise, if you make eye contact, they know you can see them, so they'll go. If you don't make eye contact, they're like,
I learned this
bicycling
at UC Davis. That's where I learned this. There are two more. Entomology prize was for a research study called A New Method of Cockroach Control on Submarines. I didn't know there was an old method, but now there's a new method of controlling cockroaches on submarines. And for the transportation prize, and the USA can be thanked for that wonderful bit of academia.
Get there? Oh, jeez.
Transportation, Namibia, South Africa, Tanzania, Zimbabwe, Brazil, UK, USA. This collaborative experiment was in order to determine by experiment whether it's safer to transport an airborne rhinoceros upside down. Should you transport rhinoceri right side up or upside down?
It's rhinoceros.
Rhinoceroses.
So what's the answer? What is it? What's the best? Because I don't know. I don't know when it's going to come up, but I'm very curious to make sure I have the right approach. If it should,
if it does come up, how to transport
like a Boy Scout, like to be ready.
They did find that suspension by the feet for 10 minutes did not impair pulmonary function more than did lateral recumbency, and apparently augmented gas exchange to a small degree relative to lateral recumbency. The biological importance in these animals of numerically small increments in pressure of oxygen, the partial pressure of oxygen, and decrements in the partial pressure of carbon dioxide with suspension by the feet is unknown. Black rhinoceroses immobilized with etorphine and azaparone were not as hypermetabolic as were white rhinoceroses immobilized with etorphine. So
does that mean however you can manage it, just go for it?
Yeah, whatever. Just if you can move a rhinoceros using a helicopter, just go for it.
What I'm hearing is if it's less than 10 minutes, and you're fine. Goes.
Yeah.
Yes. Oh my gosh. And yes, the plural is rhinoceroses or rhinoceros.
It's a bevy of rhinoceroses.
It's a crash of rhinoceroses.
Ooh, I like that.
Ones. Yeah.
A crash. Well, okay. Let's crash our celebratory symbols together for the awardees of this year's IgNobel Prizes. The Nobel Prizes are coming up in early October. Who do you think is going to win? Are mRNA vaccines in the running? We will see. We will see in just a few weeks. But in the meantime, I think it's time for Justin to tell us about how Mars rocks.
Oh, yeah. So NASA's Perseverance Mars rover has collected a second sample. And analysis of the rock samples and actually kind of the place where they took the view that they got within the rocks that they took the samples from are giving them clues already to the crater's past. They can tell there was they kind of already know there was volcanic activity and some period of persistent water. This is quoting Ken Farley of Caltech, project scientist for the mission. It looks like our first rocks reveal a potentially habitable, sustained environment. It's a big deal that the water was there a long time. I feel like NASA is starting to sort of simplify their language. I feel like they took a class. Like last week when they found the sample, they're like, I've got it. And now they're like, It's a big deal. Keep it just short, to the point. Say what the thing is. But yeah, so they've spied salts in the rock, which may indicate that groundwater flowed through and altered the mineral composition of the rock, or that the liquid evaporated, that collected there and then evaporated, leaving the salts behind. Salt minerals in these two first rocks may have also trapped tiny bubbles of ancient Martian water. Those are the things, then, if we get those samples back, we could analyze further here on Earth. If they are present, it could actually be like a time capsule for us to back engineer what processes had been taking place in the climate and in the habitat of Mars going backwards. One of the interesting things is those salt minerals that they can see already on these rocks are known to have traces and preserve signs of ancient life when we find them here on Earth. So this is probably, well, basically it just turns out we found a fantastic spot, which we kind of already knew, because the science team already knew that the lake was once filled with, or that the crater was once filled with a lake, that it had water in it. What they don't really have a solid grasp on is how long that water was present. The crater in Jazero's lake might have been, the lake and the crater might have been a sort of one-off event. There could have been a flood that filled the crater, and then it dried up within 50 years. Poof, gone. And so it has sort of the markings of what would have looked like a lake, but none of the sort of long-term persistence of 10,000 of years or a million years of being a lake that would have really allowed life to take hold and flourish there. Level of alteration that the scientists see in the rock is already providing some clues. It suggests that the groundwater was present for, and I'm quoting, a long time.
A rather long time.
Yeah, so that doesn't say tens of thousands of years versus a million years of this being a lake, but it seems to be ruling out 50 years. It seems to be ruling out the one-off event. It looks like this is a much more persistent site than just a 50-year flood and then evaporation type event. So can't quite say what has altered the rocks yet, but again, when we get them back. This is Mitch Schutley, NASA headquarters mission program scientist. These samples have a high value for future laboratory analysis back on Earth. One day, we may be able to work out the sequence and timing of the environmental conditions that this rock's minerals represent. This will help answer the big picture science question, the history and stability of liquid water on Mars.
Yeah, and I think there's some other research that came out recently through NASA confirming that there were these massive, giant volcanic eruptions on Mars, like super volcano eruptions. And so you have to kind of think about the geological formation of this planet, right? And right now we're looking at the surface. And so to know that this water and the geochemical evidence that they're finding in these surface rocks is basically, that's been there a long time, but that means, okay, how long has it been since the volcanic activity? How long has it been since it was impacted, since the planet's surface was impacted by asteroids, by meteors? So there are all these interesting questions about like the timeline of the geological events on the planet that allowed water to exist there for a little while, but then to disappear from the surface, and then also to be so different from Earth. Yeah.
To be able to analyze that water, if we get a sample of these tiny water bubbles, might answer some of the questions about how water ended up on the Earth. I mean, we have an idea that some of that water came from a comet. We also have an idea that some of that water might have already sort of been there, sort of formed on Earth. So getting another glimpse of water on a planet next door would be huge for that conversation.
Yeah, would be absolutely huge. I wonder if super volcanoes are necessary, like spew stuff into the atmosphere and cool things down. So maybe like the hot planet surface that allows condensation and water to actually stick around a bit once everything cools down a little. I don't know. spitballing a little here.
Yeah, you are, but there's also some interesting stuff about like the fact that we might have trapped samples in here, and we might have groundwater, and we might have little niches for microbial life to sort of have a little safe space to get started, is because volcanic rock is very porous. And so there's lots of little holes in there, which become micro laboratories of chemical reactions all unto themselves. So yeah, I think there is probably a very good combination of that volcanic activity and the presence of water that leads to things like.
It's a big story. It's the whole story. It all has to work together for it to happen. And whenever you're telling a story, you always have to wonder if anybody's listening. How can we find out if they're listening, Blair?
Great question. Well, as I tell you this story, I'm just gonna go ahead and I'm gonna put a nice little pulsometer on both of your fingers so that I can watch your pulse and see if you're really paying attention to me, because
I have one. I can go do this.
A recent study looked at subjects' heart rates in relation to listening to a story. And this is by themselves. And they found that heart rates synchronized at certain points of the story if people were paying attention. This is a study from City College of New York and the Paris Brain Institute. And they wanted to see how we process stimulus and if that directly relates to our heart rates. So what they did is they did 4 experiments. They took healthy volunteers in the first one. They had them listen to an audiobook of 20,000 Leagues Under the Sea. As they listened, they followed their heart rate, and it changed based on what was happening in the story. They measured with an EKG. They found that the majority of subjects showed increases and decreases at the same points in the story. Then in the second experiment, they watched short instructional videos. These are educational, and there was no underlying emotional variation, no excitement, no romance, no danger. And so that confirmed that there was emotional engagement, that it did not play a part in this synchronization. The synchronization was still happening. They were showing similar fluctuations while they were paying attention. Then they had the participants watch the same videos, these instructional videos, while counting backwards in their heads. And during that time, their heart rates went all over the place. They didn't synchronize.
Yeah, no synchronicity. Yeah.
Yes. Then in the third experiment, they listened to short children's stories, some while attentive and others while being distracted. And then they were asked to recall facts. So they found that the fluctuations were consistent and predictive in those who answered questions correctly. But those who did not answer questions correctly, their heart did not synchronize with other individuals. So that does indicate a signal of conscious processing of the narrative. Then they looked at the changes in breathing rates and didn't see any synchronization in relation to breathing rates, which is surprising because breathing directly relates to heartbeat. If you are manipulating only breathing, you'll see a change in heart rate, right? But so the breathing had nothing to do with it. And then the final experiment, they did healthy volunteers, and then they also had patients with disorders of consciousness, comas, or vegetative states. And only the healthy volunteers in that case showed heart rate synchronization. Wait
a second.
What?
They tested people in a coma? The listening ability.
Yeah.
To people who weren't in a coma's listening ability?
Correct.
How did they not get the IgNobel this year? That's what I want to know.
Well, it's a good question.
It's a new study, so maybe this next year.
You have to make sure that there's not something else going on in the room, that the sound wavelengths are affecting the... I don't know. I don't know.
Yeah,
but regardless, it was an extra level of control, I suppose.
I guess.
But all that to say, your heart rate, independent of your breathing pattern, fluctuates as compared with developments in a story or in the way you're being instructed on something in the same way another person does in a separate room in a separate part of the world. Pretty neat.
Yeah.
I
My
like to
takeaway
think,
is still,
as Fada said here in the chat, I think it's pretty awesome to think that everybody who's watching or listening to the podcast that we're doing right now, you're all synchronized. That your heart rate is synchronizing because you're listening to and paying attention to this program. So that's how we're all connected all around the world.
I think this is directly related to certain things that we were trying to do at the height of shutdown. Is we were watching concerts virtually. We were watching live events virtually. We were doing Zoom meetups with our friends, and it felt so sterilized and unsocial. But knowing this, there actually most likely was a connection in our experiences in those moments, which actually is pretty neat.
Yeah. And I think that and that connection behaviorally, socially is essential to well-being as a human, as, you know, our animal species. So these are all things that do go together scientifically. And we have been under pressure, but really cool, weird. I hear where you're going, Justin. Researchers published this last week in Physical Review D. They're from the Department of Physics and Astronomy at the University of Sussex about their discovery when they were confused about this extra number that kept showing up in equations that they were running on their quantum gravitational corrections to the entropy of a black hole. Anyway, they talked about it, they thought about it, and they realized that Schwarzschild radius black holes have pressure. Stephen Hawking hypothesized and discovered that black holes emit thermal radiation. So to have thermal radiation, that's temperature. They exert temperature, a change in thermal state onto the universe. They don't just do that, though. According to these researchers, it's not just temperature, it's also pressure. Black holes exert pressure on space-time around them. Pressure.
Yeah.
Push it.
But the thing about it is, though,
on you. What?
You're talking about, so there's a pressure on a space-time thing. It's like, still like, what does it even mean, pressure in space-time? Like, well, I don't get it.
Right. Right. What is pressure if you're talking about this black hole existing in an area that's virtually a vacuum? Not quite. Yeah. So what is this pressure? How much pressure? They're calculating this, but this quantum gravity is exerting a force through pressure. And I found this really interesting because what it does in a mental sense, and maybe this is just an analogy, of course, but as somebody who had to learn in physics and physiology about and chemistry, the ideal gas law, PV equals nRT, right? Pressure times volume equals the number of moles times R and T. And so this equation works really well for the real world, for the macro scale. And it's just fascinating to think about. There's a place in space-time where the quantum world, where we go from the singularity inside a black hole in which everything breaks down, all the equations break down, coming out of that to a place at which around the Schwarzschild radius, you have temperature and pressure being exerted. So there's a force going from quantum to actually the macro standard model influence.
Which then is a pretty solid, amazing also connection in a way, like because it says like, well, we have this phenomenon where you can do the ideal gas law and change the pressure, and then you can calculate how many molecules are going to be able to be present based on that different pressure change.
Right.
And oh, by the way, there's a reason for it. That's how the universe works all the way down. It's not just a phenomenon that has a thing to do with gases. It has to do with the actual infrastructure of the architecture of the universe. And that's why it's a law at a macro.
Yeah. Yeah. And as someone is saying, pressure equals force over area. Right. So it is a force over that area, but it's also related to the temperature, that thermal radiation at the same time. And this is, if you have the thermal radiation, it should go along with pressure. And they have figured that out. I just, it's one of those little tiny details that I think forms the connections within our understanding of how things connecting. Work. I am. I'm going to make lots of connections, lots and lots of connections.
I've got a connection to make. Yeah.
Okay. Good.
Absolutely. This is a Harvard geneticist, George Church. I think we've talked about a couple times on the show. Back 2015 and maybe earlier, talking about maybe bringing back the mammoth. He's co-founded a new company, which is sort of exciting by itself, but it's also, I think it's his like 23rd or 24th company he's co-founded. But this specific company has the goal of bringing back the woolly mammoth. And not just a one-off, not just a zoo gypsy circus sideshow anomaly. No, this is he intends to reintroduce herds of mammoths into the wild.
In quote unquote, into the wild. Like,
into the wild.
Which country is gonna go, yeah, wonderful, just let them roam free? Like, no, Russia, it's not going to be in the wild. It's going to be behind Russia. A barrier, isolated from the rest of biology.
Russia already said yes. Okay, but anyway, the company would likely wants to take woolly mammoth recovered DNA and make a hybridized Asian elephant that could actually thrive in Arctic climates, using these herds of the hybrids to convert the modern mossy, shrubby tundra back into the grassy steppes that they once were during the
Pleistocene.
Pleistocene, thank you. When mammoth's influence was strong, and this is like some 11,000 and more years ago. The reintroduction and the ensuing landscape changes could slow thaw of Arctic permafrost, which in turn could help keep dangerous methane and carbon gases out of the greenhouse economy. So this is
not taking into account the fact that it was climate change that made the woolly mammoths go extinct, not the other way around. Like, the woolly mammoths weren't there, the lone animal battling against climate change back 10,000 years ago. Nope.
Well, also, elephants are currently ecosystem architects. So the idea that you think that, or that they think, I'm not going to push all this on you, Justin, that they think that they will reduce the melting of the permafrost. Yes, fine, whatever. But they will still disturb much captured carbon as they completely tear up the area, which is what elephants do. So they're ecosystem architects because they disturb the ground. That's the thing that they do.
So that's a very good point, Blair. And I'll just leave it there. No, actually I won't.
Of course you won't.
Of course you won't.
So first of all, let me get done with Church. Church says we're de-extincting genes. De-extincting genes, not species. The goal is really a cold-resistant elephant that is going to be fully interbreedable with the endangered Asian elephant.
Because elephants do so well on the planet currently. 96 of them are killed a day.
Yeah, but this is a fair point, Blair. I mean, the first thing I thought when this announcement came through, I was like, this is a PR stunt, and this money would be better spent being put into elephant conservation.
And that's exactly what it's doing.
No, it's conserving Asian elephant genes. If it's de-extincting woolly mammoth genes, it will be preserving Asian elephant genes. So here's the question. George Church is a geneticist. He is a proponent of the gene. He is not a organismal biologist. He is not a proponent of ecosystems and organisms and individual species. He is looking at from the gene perspective. So we're going to have a big debate from the ecosystem, environment, and species perspective.
So I'll make a couple of points there. One, if you can preserve the elephant but make it so it can live in a much colder habitat where there's less people and poachers and can be protected, you're preserving a species to an extent. The other
You're
thing
assuming that Russia will be able to protect these mammoths and that they won't be hunted for sport because, oh my goodness, could you imagine having a de-extincted mammoth hybrid elephant in your head on your wall? Oh my God, they are going to be hunted for sport.
Okay, and maybe
that's what's going to happen.
And maybe for food. But let me ask you this.
No.
Let me ask you this.
For food, that's too expensive. This is a business. This is a startup. This is a venture-backed startup. This is not going to be, to help people for food. No, this is all the money. All they care about is the money. And George Church is just like, Gene.
Okay. So according, let me get finished with Church. I didn't get done with Church. Church says that the first hybrid calf could be 6 years away. A self-sustaining herd might take decades. So let's move on to Sergei Zimov, who is Sergei is a Russian ecologist who back in 1996 started working on the Pleistocene Park. This is a fenced-off area of the tundra in northern Russia. He's introduced elk, bison, reindeer, some kind of camel, some other large herbivores to test the creature's effects on the landscape. So what he's found is, yes, they can have this sort of effect of moving seeds around, increasing, eating up the shrubs and stuff. And it's mostly stuff is now shrubs, trees, and mosses. Back in the day, those were all knocked down by elephants, like you say, tearing stuff up, mammoths tearing stuff up, knocking stuff down. And was replaced with, it was grasses that were there because those grass seeds were ending up in the mammoth and they're getting deposited with fertilizer and they're growing everywhere. Okay. One of the things that he discovered there is that the act of these herbivores going around and knocking down the ice made the cold of the northern tundra penetrate deeper into the tundra itself and made it colder, as much as 10 degrees colder than areas that were not affected by these wildlife. So his idea is, hey, this is a perfect solution for keeping carbons and methanes in if we can keep the tundra 10 degrees colder than it is now. Because the other thing, other than occasionally knocking down trees and tearing stuff up, the other thing a mammoth would do would be pushing big holes in the ice everywhere it went. Sort of like how they oxygenate a lawn by wearing spikes on shoes and like walk around to get the, I don't know if that's a real thing, if it works, but people do it. By knocking down the ice, by compacting it, they actually allow the cold air of the Arctic to penetrate deeper into the soil, freeze it more thoroughly, and prevent the thaw of the tundra, which is one of the things that they're looking at. The other thing Church's lab has been doing, though, is they have also been working on ways to, they don't plan to use an endangered elephant to bear the mammoth child. They're actually working on the thing that, remember a long time ago, they were trying to see if they could rear sheep in these bags. Do you remember this story?
The lamb bags. Yeah.
The lamb bags. They're going to try to pursue a mammoth bag version of this. They still have to figure out, they're going to want to socialize probably with the Asian elephant when the thing is born. They want to socialize it with the first generations, at least with some other elephants. But they're not going to use them as proxies there. If this is successful, it then means they can use that technology to approach any other endangered animal. So you're talking
and that's
rhinos,
the interesting thing. So
elephants, you're talking
if they can use it to bring back, I'm the woolly mammoth. It's a PR stunt. So but if it can kickstart more funding for these technologies to bring back species that humans forced extinct recently. White rhinos,
Thylacines.
the very recent extinctions. But at the same time, we need to consider what the Earth is like now and whether or not we can maintain the species that are here, the genes that are here. I get excited about this technology and the idea of doing it. And no, it's not Jurassic Park. It's not even necessarily going to be a Pleistocene park, but the attitude that presages these advancements is one of a human ability to ultimately affect the environment on the Earth and to be the ultimate master of this planet. And I think there's a lot of hubris there. But who knows?
Well,
maybe I'm wrong.
But we already have these problems. We have species going extinct at record numbers every year. We have a need, if we want, I guess it's not a need, it's a choice to preserve and to try to rebuild populations. And I think the woolly mammoth, with this global warming, with this additional positive effect it has on keeping the tundra cold, and perhaps those grasses can bind and sequester as well, I think that's a good enough story that I'm in. I mean, I wasn't in before, but now I'm in for better reasons than I had before.
Many more reasons. But maybe you should listen to your spirit animal, Jeff Goldblum.
My uncle? What's Uncle Goldblum says? You were so preoccupied with whether or not
do the
you
voice.
could.
Don't just read it. Do the voice.
Yeah.
Come on.
You were so preoccupied with whether or not you could. You didn't stop to think if you should. Pretty good.
It was acceptable. That's all right. All right, everybody. If you've made it this far, this is This Week in Science. We may or may not have more debates ahead, but we definitely have a lot more science. If you're enjoying the show, please share it with a friend today, right now even. Hit up that social media. Tell everybody you're watching or listening. Share TWIS. All right, time to come back for the COVID update. Let's try and run through some of these things. COVID update.
I know it's not good.
Oh,
No.
actually it is.
No.
There's a lot of good news in the COVID update. So
I had
let's
to put my
start
good
with
news
the bad
segment.
news.
The Just Good News segment had to go into the COVID section this week.
Okay, so you're teasing everybody that there's some good news to come.
Oh, yeah.
Well, let me tell you the bad news.
Start there.
I'll do that. I'll start there. According to the CDC COVID tracking, 1 in 500 Americans has died from COVID-19.
Mm-hmm.
And it's actually more than that.
Wait, what?
Mm-hmm.
Yeah.
That's-
Yeah.
Mhm.
1 in 500 Americans has died from COVID-19.
Yeah.
So just taking that statistic for the reality of the numbers of people who have died at this point in time. Many of them more recently, very preventably with vaccination. Now, into interesting science about SARS-like viruses and things like COVID-19. There is a study out this week reporting that SARS viruses make the leap from animals to humans much more often than thought. Researchers overlaid the distribution of bat species and where humans live, mostly in Southeast Asia, to infer the potential overlap of bat species and who, as we know from previous interviews, specifically with Angelique Cortals and Liliana Davalos and their collaborators, that these bats carry a lot of viruses and they're reservoirs for viruses. And the question is, how often do these things spill over into human populations? This paper estimates that there are approximately 400,000 human infections with SARS family viruses every year. So the big question is, what makes it out of those 400,000 some odd cases? What is it that makes a virus really take the jump and go from a more localized outbreak to something that becomes a large pandemic?
I think it takes one extrovert.
Could be an extrovert. There are many, many factors in place there. But if you're interested, the research of our guests, Angelique Cortals and Liliana Davalos, is actually featured in a PBS special on bat superpowers that is online now and will be airing on PBS stations across the United States very soon. I don't think online it's available outside the United States, unless you have a VPN.
Oh, I've got to get one of them.
Yeah. Anyway, another study, a couple of studies that are out this week related to fertility. I don't know if you saw Nicki Minaj's tweet this week from the Met Gala, where usually she would be there wearing some fabulous outfit, but she didn't go this year because she's not vaccinated. And she said she couldn't go because like a friend of her cousin got vaccinated and then ended up with like swelled up testicles. And then his girlfriend dumped him. And so, of course, she can't go to the Met Gala because she couldn't get vaccinated and she needed to finish doing her own research.
Okay, well but that makes sense because you're supposed to get the shot in your arm, right? If you get it in the wrong place, bad things can happen. I would agree. But caution against that.
You want to get your if they're trying to give you the vaccine elsewhere, you should probably talk to them, question what they're doing. These studies out this week confirm the vaccines. None of the vaccines that have been approved in the United States or elsewhere that they have around the globe have been seen to affect male or female fertility or to swell the testicles. And COVID infection can actually do all of the above. So it's a better bet to get vaccinated than it is to get infected by COVID if you are worried about your fertility. Additionally, though, it seems like a lot of people around the world, United States and in Europe, are not thinking about fertility, reproduction in general. People are not reproducing in Europe and the United States. But I guess, if you're not sleeping, you're stressed out, you're not very happy, it doesn't really put you in the mood for babies.
Didn't go on an online date for almost 2 years. There's lots of things that stop it.
All right. I have some good news, but Justin, I think I'd like you to start out some of this COVID stuff because you had a bad news and then a good news story.
Yeah, I guess that this is COVID. Actually, maybe like Menage. I don't know. Isn't that pretty after? Turns out, COVID has been using a social filter on us this entire time. This is a study the Instituto de Radio Televisión Española and the UAB conducted during the COVID-19 lockdown. They found that black and white 2D images of SARS-CoV-2 make the virus seem more infectious. And it's revealed that that's the only images we have are black and white 2D images that scientists have been able to produce. The three-dimensional, multicolored, multifaceted, high-resolution images that we keep seeing over and over again with every news report aren't real. More so, when they show the virus as that sort of beautiful, not really realistic thing, people are less afraid of it. There's a connection between seeing that version of it versus the real, like, hey, this is a totally tiny thing. It's hard even to see or detect. Here's some horrible, bad resolution, black and white images that can kind of show a virus from a bit of an angle. Those were terrifying. The one that makes it look like it doesn't have any wrinkles, no dimples on the backside, no cellulite.
Right. Yeah, it's the pretty face filter.
Yeah, the pretty face on COVID. Makes sense, when they give the COVID vaccine those anime eyes, that's when you really know they're gone too far. Yeah.
Kawaii. Okay. Yeah, it's interesting to have people talking about how COVID is presented in the media. We want pretty pictures, but yeah, they're limited. So then they have to be spiced up and spruced up and and we forget how much that affects people. So yeah, that's a really interesting point. Oh, on this podcast from here on out, only the ugliest COVID pictures.
Yeah, just show
only the ugliest ones. Give me some
let's
good
not
news.
call it ugly. This is just show it for real. Let's just keep it real.
We'll keep it real pictures of COVID. That's right.
Yeah. We don't want young viruses aspiring to an unattainable role model. I guess it's time for Just Good News, everybody. The segment that takes the worst, horriblest, most awfulest thing you don't really want to hear anymore about topic in science and looks for the one good story that makes it sound a little better. COVID edition. New study from La Jolla Institute for Immunology helps answer a question: how long is this immunity against COVID-19 good for if you get vaccinated? They report in Science, a low dose of Moderna vaccine lasts for at least 6 months. And there was no indicator that vaccinated people will need a booster shot. When you hear it only lasts 6 months, or you hear it lasts all the way up to 6 months, you might think, oh, well, then of course you need a booster in 6 months. But that's not really what this report is saying. First of all, backtrack a little. They didn't test the vaccine in the way that people have been getting it. They tested it the way it was initially tested in the first round of studies, which is a quarter of the current dose. So they did this quarter dose, one quarter dose, and they compared it to people who had gotten COVID naturally. And they found that it was really not a big difference. People who had gotten COVID and recovered, not the people who died, obviously there's a difference there. People who had gotten COVID and recovered from it had the antibody immunity of somebody who had gotten one quarter dose of the Moderna. And what they also found was at that 6-month period, the body's immune system had picked up and continued its sort of knowledge of how to handle this virus. The immune system got trained by that 6 months where the vaccine was being effective, which means there's a persistent immunity quotient that becomes available. And again, this is comparing people who had gotten COVID and the natural response that the immune system had with a quarter of the single dose that people in the initial trial, the very first way back trial, had encountered. So what we have now are people getting these 2 doses, 4 times the amount, and spread out because we found this sweet spot. So they don't know. They say okay, we can't say that the vaccine in the quantities that people are getting now are much more effective. Might expect it to be, but we didn't actually. That's not what they studied. But what they did show was a staying power for an mRNA virus that was training the immune system to take over and handle things on its own. And this just in, there's another story of more good news.
I do have more good news. So Blair was talking earlier. Happy good news. Yeah, so all right, boosters, everybody. Long-lasting Moderna. Very interesting. We'll see. The data is yet to come. There are competing studies out there, dueling studies, preprints and postprints and all of the above, but just get vaccinated. So there's the big question, and we're on social media and even looking at the media these days, it's hard to believe that that it's not a majority of people who are fighting against mask mandates or social distancing or these restrictions that have come about as a result of this public health emergency, COVID-19, that has killed 1 in 500 Americans. Pew Research did a survey, and they found that 73% of their sample set said they were vaccinated. At least half of those people expressed confusion, concern over vaccine information and health impacts. But a majority of people in the United States agree that the costs of the restrictions that we've allowed into our lives were worth it. There are downsides and people see them, but the majority of people agree that there's a benefit. Now, there's a huge partisan gap, which you would probably expect, but majorities in the United States support proof of vaccination for air travel and college. 58% of Americans express concerns about unknowingly spreading the coronavirus. We know asymptomatic cases can be super spreaders. Americans, it's very rare, however, Americans who are vaccinated and not vaccinated do see COVID-19 vaccines in very different ways, which you would expect based on what we've been talking about over the last 18, 19, 20 months. Americans hold mixed reactions to changing public health guidance during the coronavirus outbreak. Don't wear masks. Wear masks. Get better masks. Oh, don't social distance. What is social distancing? These new words we had to learn, these new behaviors. Things were changing, but a lot of people understand that that is the public health infrastructure following the science as we learn more. Few Americans know access to vaccines is limited in developing countries. So there's kind of this expectation here in the United States that it's like it is here everywhere else, which it is not necessarily. But this is a really interesting survey with the big bottom line that 62% of people surveyed said that it is worth the cost, that saving people's lives is worth the restrictions, even though there have been negative effects on businesses and economic activity. And
I think this feeds itself.
People self-anchor the answers.
The people who see this as an attack on their freedoms probably also don't understand that they are in the minority. It's part of a false kind of equivalency of it's the whole conversation that we've had a million times about climate denial that John Oliver did so well, where he brought on 97 climate scientists to talk about climate change and 3 climate deniers to talk about how climate change wasn't real, right? And that's actually the debate, though you see 1 and 1. And that's exactly what was happening here. Is there was an understanding that for every one person that believed in science, wanted a vaccine, and supported the lockdown, we were presented with one person who didn't. And that was not actually representative of what was going on.
Yeah, and it's not. And so there is the false equivalency in the media and in social media where people are louder, where people can be magnified in their impact, even though they have a minority viewpoint. It's worth checking ourselves every once in a while and also reconsidering our perspectives. And the data within this survey does also remind us that it's not black and white, that there are a lot of different perspectives, even within the partisan divide, even within the vaccinated, not vaccinated divide. It's a very complex situation. Each individual is a microcosm of their experiences and complexity.
Yeah.
And the big thing is, though...
But there's also the ability for people to just get influenced. And by nonsense. Because... I bet most of the people who are arguing against getting this vaccine have 5 or 6 or 7 vaccines in their medical history that have never affected anything.
Many of them do.
Yeah.
And so to the point of Nicki Minaj wanting to do her own research, other people wanting to do their own research, even people who are vaccinated and came around to the idea of becoming vaccinated against COVID, a lot of people, it's 50% of vaccinated, 70% of unvaccinated say it's hard to make sense of all the information about COVID-19 vaccines. So science communicators, if there are public health science communicators out there, we have our work cut out for us to try and make it. It's obviously a problem. We are not getting the information across accessibly. And that is not necessarily on the receiver. Oh my goodness. Okay, no more COVID news. Let's move away from COVID. This is This Week in Science. It's time for us to move on in the show and get back to science-y science, not COVID-y science. So let's discuss you becoming a supporter of This Week in Science on Patreon. If you head over to This Week in Science's website, twist.org, and click on the Patreon link, that'll take you to our Patreon community page where you can choose your level of support, $10 and more per month, and you will be thanked by name at the end of the show. You'll also get some goodies in the mail at some point, and we all love getting things in the mail. It makes us feel pretty special. You can be part of the TWIS Club. That's right. The club of TWIS supporters. Know that you are one of the people in this world who are helping to bring This Week in Science to more and more people. So head over there right now. We can't do this without you. We really appreciate your support. And now it's time to come back with more This Week in Science and that wonderful part of the show that we love and we enjoy because it's bloodthirsty. And, oh, it's Blair's Animal Corner.
With Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
Oh my goodness. Well, you know how you have your pet cow and just, darn it if she doesn't wreck the carpet again. It's really
I know.
time.
You let the cow in the living room and
just, you say,
Bessie, what you
this
doing
once-
over there in the corner?
This once you can sleep in the house. Oh, nope. Problem again. Got me again. So toilet training cows. Let's talk about this. Is a kind of a proof of concept looking at the fact that cow urine is really high in nitrogen, as much urine is. And this contributes to a range of environmental problems, mainly looking at water contamination and climate change. So the nitrogen in urine breaks down in the soil. This produces nitrate and nitrous oxide. Nitrate leaches into lakes, rivers, and aquifers. It pollutes water. It contributes to excessive growth of weeds and algae. It's bad. And then nitrous oxide is a greenhouse gas. It accounts for about 12% of New Zealand's greenhouse gas emissions. Of course, it's, don't get me started on the red herring of other gases related to climate change besides carbon, but that doesn't mean we can't do both at the same time, right? Carbon, I'm still going to say, it's going to be on my tombstone. We should have followed the carbon. But this is still also important. We can do two things at once. So anyway, all that to say, that would be great, not to mention the fact that I'm sure when you have cows on pasture, if they are urinating on the pasture all the time, that's probably not good for the pasture itself as well, which probably doesn't positively impact the cows' lives over generations. Anyway, point being, there's a lot of reasons that having cows kind of cooped up into one location causes all sorts of environmental problems with their urine. And so that's, in New Zealand and Australia, they're outside all the time. These things happen out on the pasture. In Europe and North America, a lot of cows are kept in barns, and then that actually accumulates ammonia. That's when nitrogen from urine mixes with feces on the barn floor. So, all that to say, cow pee is a problem. So if it could be captured and treated, that would be good for lots of reasons. This research project looked at applied principles from behavioral psychology, the other PS, to train young cattle to urinate in a particular place. They essentially toilet trained them. And they actually used very similar methods to toilet training children, to potty training other animals. They call it backward chaining. So essentially, in the animal training world and in kind of the zoo world, also this is called kind of catching a behavior. So they were confined into the toilet area. They were just kind of waited around this latrine pen, and they were reinforced with a treat when they peed. It was like, yay, you did it where you were supposed to do it. What a good job. And this established the pen as the place where they urinate. If they urinate in the pen, they get a treat. Perfect. Then they were placed in an alley outside the pen, still nice and close. They were reinforced for entering the pen and going to the bathroom. If it began in the alley, they would get a short, small spray of water to kind of like jog them out of it. And then they would walk in and they would pee in the barn. So 7 out of 8 calves were successfully trained, and it took about 15 days of training. The majority learned the full set of skills within 20 to 25 urinations. So I don't have any children of my own, but I'm pretty sure that's very fast compared to toilet training children, which they did say that is considerably quicker than toilet training a 3 or 4-year-old.
Well,
it really depends.
Yeah, it's sort of like when they're ready to be trained, like when they're ready to take it on, it's usually pretty quick.
Well, you're adding in the fact that human babies can also understand some speech, so you can also kind of reason with them in a whole different way. But it doesn't matter. But anywho, they were able to toilet train these cattle, these 7 of 8 cattle. So that means that, first of all, they could associate a certain area with a place they were supposed to go, but also that they learned to attend to their own urination reflex. They were able to hold it, essentially. I'm not surprised by that, because they're mammals. And we train cats and dogs to do this. Not to mention, there are lots of animals that you can potty train or that latrine on their own. Rhinos, we were talking about rhinos earlier. They like to have their very own midden. I always say that wrong. Midden. It's like a poop
It's a bathroom
pile,
for rhinos.
Yeah, it's a poop pile. So lots of animals do this on their own. They have a designated place they like to go to the bathroom. So it is also possible, I will just throw out there, maybe cows have wanted a latrine this whole time. And you're like, I don't know, pee on your grass. I don't like this. So well, just throw it out there.
But I mean, I think part of this story just does say that people have underestimated cow intelligence or trainability.
Yeah. So anyway, the next step of this research is to try to scale it up. So they did this with 7 of 8 cows. So the idea is for this to actually do anything for the environment, you'd have to get this going on a much larger scale. You can tell small family farms to do these kind of environmentally friendly practices all day, but really you need kind of those large scale farms, dairy farms, meat farms, to do this properly in order for it to actually make an impact on the environment. So what they suggest would be the next step would be a way to detect urination, because they did this by watching the cows, and then also to deliver reinforcement automatically without human intervention. So basically they could set this machine up, and I'm already picturing it's called a Furbo, which is like a little treat dispenser that you can buy for your cat or your dog that you can use on your phone, and you can dispense them a treat when you're at work. I don't have this, but I think it'd be very fun.
That's pretty smart.
But you could have something very easily that senses urination on this platform and then dispenses a treat. I think it would be pretty easy to do. But so that would be their next step, would be to try to automate this so that you could train cows en masse to do this. Yes. Yeah. Okay, so the last thing that they want to do is figure out how far cattle are willing to go to use the urination stall, which reminded me of the famous research that they did at Disneyland to figure out exactly how many steps a human will go carrying garbage before they drop it on the ground. So there are trash cans every, I think it's 75 steps in Disneyland, because they figured out that's exactly how far the average human is willing to carry a piece of garbage before they litter. Yeah, but so similarly, figuring out how far cows are willing to walk holding it before they will then just pee on the ground. So this is an important piece of the research to figure out how to incentivize the cows to do this in large bits. But I love the idea of using behavioral conditioning to improve the welfare of animals in farming and also to help with the environmental impact. Very cool.
Right. I like the idea of incentivizing cows. What is an incentive for a cow? What do they really
A treat.
want? They just want a treat. Yeah,
Just
they
want some
do.
treats. And then
Nice.
from toilet trained cows to cannibalistic butterflies. So everyone loves a butterfly. They're beautiful. They're so fun. When you love to have a butterfly just land on you, majesty. Well, not if you're a caterpillar of the same species. This is a study from the University of Sydney, and they found that milkweed butterflies have been observed feeding on live caterpillars of their own species. That is their own species young.
But they got a proboscis? Is that what it's called? It's just a little like straw. It's just like
proboscis. It's not like a proboscis. No, it's a proboscis.
Proboscis.
Yes, I will tell you all about the mechanics of this grisly demise. So the milkweed butterflies, they like to eat when they're caterpillars, when they're still babies. They feed on toxic plants. They hold those chemicals. They sequester them for self-defense in their bodies. Those chemicals are noxious. They are unpalatable to predators. So, for example, birds that would otherwise eat caterpillars are deterred by these toxic plants when they're both caterpillars and when they grow up and metamorphose into butterflies. And so they also partially responsible for the bright colors of butterflies. That is warning colors saying, I am poisonous. So they're munching on this when they're caterpillars. They are accumulating all of these chemicals. And so they also, the third use of these toxins are to help male butterflies make mating pheromones. And they also then package those into nuptial gifts to females during courtship. So all this to say, without these toxic plants, caterpillar and butterfly, and then future caterpillars don't exist. And so
they need the toxins. They need all the stuff that the milkweed gives them.
Yes, absolutely. Yeah, and so the male butterflies, after they're older, after they've already metamorphosed out of caterpillar stage, they will supplement their diet by looking for more resources of those chemicals. And they are usually observed getting them through plant sources. So the way that they do that is they actually damage the plants. They scratch them with their sharp tarsal claws. Then the plants kind of start like almost bleeding. They start releasing this juice, and then the butterflies.
They're milking the milkweed.
Yeah, they can lap up the little chemicals straight from the plants, from like the plant blood, using their long curly tongues. Caterpillars, all they do their whole life until they metamorphose is eat these leaves. And so caterpillars, in the words of some of these researchers, are quote unquote bags of macerated leaves. These are the same leaves that the butterflies are scratching at to suck this liquid. And so adult butterflies were observed scratching at both live and dead caterpillars, tearing them open to imbibe their juices. The caterpillar larvae, they would contort their bodies rapidly. They were trying to deter the scratching. And butterflies, they were just lapping up all of the juices they fed on the carcasses of their very own species of caterpillars. So
it's easier. Is the skin of a caterpillar thinner, thicker, more delicate than the cuticle, I guess it would be, of a plant?
Great question. That's an excellent next
question. Is it easier to access the plant bags?
Or are they desperate? Or are they doing this all the time and we just didn't know? Is this the first time in 2019 in North Sulawesi in Indonesia that they saw this happen? Is this the very first time? And it's just always been part of their deal. But the other crazy thing here is that this behavior does not fit in the traditional models of evolutionary strategy of predation, parasitism, or mutualism. And so they have actually coined a new phrase for this, which is kleptopharmacophagy. Chemical
stealing.
For consumption.
Yeah, pharma is the chemical or the pharmacological. Pharmacophagy?
Phagy, yeah, is eating. Consumption. Yeah.
Consumption, yeah, okay.
Kleptopharmacophagy. Kleptopharmacophagy, probably. Kleptopharmacophagy. Yes. So this is a new idea because evolutionarily, it would not be beneficial for them to kill their own young, to rob them of chemicals, to then give birth, to lay more eggs that would result in more young, right? That doesn't make any sense. So this is a whole new thing, crazy. They want to see if they can find more of it. They want to find out exactly what compounds the butterflies are interested in. They want to find out if other butterflies in other places of the world do this.
Yeah,
So
well, I wonder how
lots of
far do the
questions.
butterflies fly away from where they pupate? So are the caterpillars in a different location from where the butterflies originated? And if so, that would necessarily mean that they're distantly related or not related. So that would potentially mean that they are getting rid of competition for resources, and it's also easy access to food and energy for their own survival, for their own reproduction.
Yeah, and that could just be it. But if one day we find out that
seriously,
8-year-old blood actually will make you live another 50 years as an adult, you gotta hide all the 8-year-olds. Like, you'll have to hide them all because people will start taking their blood.
They'll scratch them and lap it up.
That's no good.
Like once that secret's out of the bag, it's just going to be trouble for everybody. But
wow, cannibalistic butterflies. I am never going to look at these butterflies the same again. Butterflies. You don't know what they're up to.
No.
They're not all sweet and pretty. Fluttering around. Oh no. Wow, that is this. This news has me shook, Blair. It really does.
Just don't be a caterpillar. You'll be fine.
I'm not a caterpillar. I think I'm going to be okay. This is This Week in Science. We are going to move into our last couple of segments of stories. I hope that you're enjoying the show. If you want a Zazzle beach towel, you should head over to twist.org and click on that Zazzle link. You can find the beach towels. They are very cool. I have one. I really like it. Really, really like it. A mug, a lumbar pillow. So many cool things. Head over to twist.org and click on that Zazzle link. All right, Justin, it's your turn.
I want to see the mug that can also double as a lumbar pillow. That sounds really interesting. Last story of the night for me. For those who are of a certain age, and were once kids, you may remember an action doll called Stretch Armstrong, or his equally stretchy nemesis, Stretch Monster.
Monster? I don't remember the monster.
I think I had the monster. I didn't have the Armstrong. I think I had the monster. He looked like lizard people, kind of almost Godzilla-ish, but more human-y form.
I had his dog.
But these were rubbery, gel-filled toys that you could just pull the arms really far apart, and you could tie them up, and then they would sort of slowly retake their original shape. They would come back. And if you're like some kids of a certain age, you may have eventually gotten bored with the stretchy toy and gotten curious to see what was inside. Might have used a proboscis or a proboscis. Whatever, you might have cut it open just to see what was inside, and quickly found out that there's this goo that just starts oozing out now when you stretch it, and it no longer becomes a toy that you can keep. So the question is, why don't humans just ooze out when we get a cut? What's the thing that keeps all of our cells from just sliding out whenever we get a minor injury? And yes, there's the blood that's coming out, but that's actually doing the clotting thing that's preventing it's sealing the wound. But the answer, turns out, is tiny anchors. Most of the cells in our bodies, whatever they are, bone, muscle, organ tissue, are locked into the right place and don't just squish off with the help of tiny anchors. These are called focal adhesions. These are strong anchors that use protein chains to link the cell to collagen, which is a protein that gives the structures to our body. These little tiny, super tiny anchors, and what are we talking about here when we're saying tiny? These are 1/10,000th the size of a human hair, few nanometers thick. That's all they are. They're just super tiny, and they've only sort of just been discovered and imaged and understood. This is what they're now looking at is, now that they sort of know this structure is here, now that they know these tiny filamental anchors are here connecting things together, they now are starting to look at what happens when those break down. And the first thing that they can see when those things are not present is when cancer is taken. Cancer is sort of runaway growth. It's also metastasization, which means the cells that are in that cancery growth spurt tissues don't anchor. They go, I'm not attached. I'll just go play the field. I might jump in the bloodstream, or however I get there through a lymphatic system. I might move over to somewhere else. And you know what? Now that I'm here, I'm going to start growing another tumor, because that's what cancer likes to do. So this is a whole, all of the things. It's one of those things when you have to okay, now we go back and rewrite all of the textbooks, look at all of the other studies, and now we have a factor, a cofactor, a thing that we didn't know was part of what was taking place that we now can go and look at and see how it fits into the whole ecosystem of proliferation and metastasization of cells. So really fascinating study. They used the cryo-electron microscope for this, which is a microscope that actually got somebody the Nobel Prize in chemistry in 2017. It's a very newish and very award-winning, but it's the only way they could even see that this thing even exists. So, whole new piece of how anatomy is put together. We were talking about pressure in the black hole.
Right.
That maybe explains all of the other levels of pressure above it, even if it's present at the quantum level. If it's now we're finding anchors and attachments in the human body that we didn't know existed previously. Another whole way of looking at the human body and human tissue interaction then becomes available.
Yeah, and it's really interesting to be thinking about how they interact in such a way that we can predict certain behaviors. So like a mutation that changes the adhesion that leads to the cells metastasizing and just taking off and going, I don't live here anymore, and running away. I'm not going to hold on to my neighbors anymore. I don't care about that. It's time for me to go do my own thing.
And if you can adjust that, right? Then if you can target that and say, now I'm going to make these mast cells stick
somewhere. Yeah.
Then you can do an excision and remove, as opposed, right? So it opens up a whole new level of treatment for cancers.
Control and treatment.
As this future goes forward, as the research.
That's super cool. That's really cool. Anything that we can do to reduce the damage done by so much cancer in our society. We're getting better and better at treating it. People are able to live with it longer, able to do so much, but we can just get rid of it. Let's figure out how to control it. Go away. Like aging. Don't we want to control aging? Blair, you're ready for this.
I'm so ready. I'm going to take notes.
I just want to keep the kids off my lawn.
So a new study out this last week is looking at protein production as a factor of aging. We know that proteins get kind of weird when we start aging, that proteins start, they don't work quite as well anymore, that the transcription and the translation is less accurate. And there are genes that are responsible for the process of transcription and translation and maintaining accuracy. And there is a particular protein and gene that were looked at in this study that was published just this last week in Cell Metabolism. Researchers were interested in how errors in protein creation correlate to lifespan, to longevity. And so they ended up diving into this one particular gene and its protein to see how it affected things. They discovered that this gene, RPS23, is so critical for translation accuracy that there's at least one part of its sequence, its genetic sequence, that is basically unchanged. It's been conserved across pretty much every species that they've looked at, except for those extremophiles that like to live in really, really hot environments. And the extremophiles that live in really, really hot environments, they have this single nucleotide mutation. This one nucleotide, this one base mutated. It's a little different. And so the researchers were like, okay, I wonder what would happen if we took the mutation that's in these extremophiles who live in these hot environments, and we put that mutation into the gene that's in every other species. If we just made that switch, what would happen? So let's look at yeast. Let's take a look at Drosophila melanogaster. Let's look at yeast. Let's see what happens with C. elegans. What happens? When they changed that one single nucleotide mutation, they found that the organisms lived longer. Their longevity was increased. Their proteins were translated with higher fidelity. There were fewer errors. But that sounds like really great news. But some of the odd things that occurred as well is that along with that increase in longevity, the developmental period also expanded. So it took longer for these organisms to get through their immature stages to their mature stages. Everything kind of took longer. But that was okay because they lived longer.
Okay, but then what I immediately want to know is if there's a gene that makes them get past that mature site faster, or gets to the mature site faster. And who cares about longevity? Like if you pull it out completely, do they because that's usually what you want in a biotech environment is you want something that'll grow really fast.
Oh, well, okay, biotech, you want it to grow really fast. But in this particular situation, they're thinking, we just made flies that live longer. What would happen if we put this in mice? If it works in mice, what's going to happen if we change it in humans? So the question is, how will it affect complex organismal biology, physiology, and is it negative? Because the trade-off that they're thinking is that maybe it wouldn't, in all these species, other than these really hot, environment-loving bacterial species, what would keep it from mutating? Why not have more longevity, right? Why not have higher fidelity? Why not have better translation transcription? Why would this gene not mutate? Why is it so conserved? And they think it's because it's the trade-off for getting to adulthood faster is that you're going to be more successful and have more offspring. And so in just a normal natural selection process, longer developmental periods are not beneficial.
Well, in any life, because you have less opportunity to die before you reproduce.
Right.
So not to mention, this extending life or is it extending reproductive lifespan? Because if it's just extending life, then there would be no evolutionary reason for that to happen. Really?
Oh, absolutely.
And that's why we got to look for it, is because it's not going to happen naturally as much, because if you are no longer reproductively viable, why would your genes mutate to make you live longer? They wouldn't. Yeah. You're taking away resources from your future generations, essentially.
Yeah.
That's true maybe for most life, actually. So it's probably true. But there's when you look at bonobo, baboon,
yes, there's
elephant,
the grandmother hypothesis.
Culture,
yes,
there's grandmother hypothesis that extends life. Yeah.
But there's only so long,
Yeah, and to all of this, there is the question also, do you live longer, but do you have the long, slow decline, and do you start feeling poorly early? I mean, how does that longevity work? The flies they're geriatric flies were still very youthful longer, when similarly aged control flies were just not very active. So They lived healthier longer. And the big thing is they could deal with higher temperatures, which, hey, maybe that's a good thing with climate change.
Thing we need.
Yeah.
We need that.
But it also means your kids are going to be at home and well into their 40s.
Yeah. And I have a couple of two quick stories that I just want to wrap with the idea of we are advancing technology at this point in time that is going to allow us to fix so many problems. And so many studies recently are like organoid this, organoid that, and we can make organoids and study all this stuff. There was a pancreatic organoid that was created this last week that they're going to be testing pancreas science on, looking at drugs for insulin resistance and all sorts of things that could help diabetics. Another study that's out this week, it created cochlea organoids. So your inner ear. And as I'm sitting here, I'm not really hearing the high-pitched, there it is, the high-pitched that's in my ears constantly because I have tinnitus or tinnitus, however you want to pronounce it. That is a sign of hearing damage and hair cell loss. My ears are damaged because I was dumb and went to loud rock concerts and really enjoyed that stuff in college. Blair's raising her hand as well. And maybe many of you out there, depending on your age, are experiencing similar impacts.
I'm sorry. Can you say all that in a lower frequency? I heard none of.
Right. You lose that higher range of hearing as you get older. Yeah.
You're like, and it makes it sound like, and then there was silence.
Right. But if you maintain the health of your hair cells in the cochlea, in your inner ear, it can allow you to maintain your hearing into old age. But what happens is when you damage your hair cells, they're damaged, and a lot of damage over a long-term period of time, they die. And that's what leads to this hearing loss, the tinnitus that I'm dealing with, and all sorts of not being able to hear higher registers, all sorts of stuff like that. So what if we could bring back hair cells? What if we could regrow them? We've talked with Allison Coffin on this show previously about hearing loss and hair cells, and she studies fish. In fish, they can regenerate their hair cells. People cannot. Mammals cannot, for the most part, regenerate their hair cells. What if we could? And so this group of researchers has developed a cochlear organoid in which they are doing a large-scale drug screen to determine whether or not there are any targets and also any drugs that we can use to regrow hair cells. And they actually discovered one cancer drug, anti-cancer drug, that is approved by the FDA for use against cancer, that through a different mechanism altogether, it stops blood cells from forming, so stops cancer from developing. But in this case, it's a completely different mechanism that allows it to, it blocks an inhibitor of cellular growth in the cochlea. Could it be used? Yeah, could it be used to bring back hair cells and restore hearing health? They're starting to do animal studies on that now.
So, vascularization reduces the production of the hairs? Is that
no, it's not the vascularization. It's completely different mechanism. So it's in cancer, it stops the vascularization. This drug that they've discovered works in the hair cells. In the cochlear organoid, it's not to do with vascularization, but there's a molecular signal that it blocks. And that molecular signal blocks the ability of the hair cells to regrow. So when you take away that break, the hair cells are able to regrow. At least they showed that in the organoid. Now we have to see if it works in animals. Maybe it'll just work in mice. I don't know. We'll find out. But this is a very exciting development as a platform for learning about potential treatments and potential targets. And my final quick story is a similar technology. the KidneyX Prize. There is a prize between the U.S. government and the American Association of Nephrology that are trying to develop a synthetic kidney that can be implanted to help people who have kidney disease, who are on dialysis, who are waiting for kidney transplants, who, have all sorts of loss of the good factors of life, right? So having to be on dialysis and on a dialysis machine, if they could instead implant a synthetic kidney-like device, they might be able to do away with dialysis altogether. A UCSF team has created a device. They just won the first of the KidneyX Prize to get $650,000. It's a drop in the bucket when it comes to technology like this, but it is going to support the development from a prototype to clinical trials to see whether or not this device, this bio-artificial kidney, can work in people, and they think it will. So anyway, there are 6 other teams that have also received awards, but this one is very promising. So congratulations. Maybe we will have devices to do away with kidney transplants and dialysis machines. Maybe we will have drugs that will bring our hearing back to our youthful levels. Maybe we will scrape the 8-year-olds and lap their blood. No, I'm kidding.
No. But yes, even if you could improve hearing so that it was better, I don't need it to be perfect, but also just if the artificial kidneys could just reduce how often people have to go do dialysis, it would be a huge increase in quality of life. Yeah. So, we can shoot for the moon. I love that. Like, oh, what if we could just give you an artificial kidney and you don't have to do dialysis anymore at all? But even putting kind of a Band-Aid on it to make that process have to happen less often would be a huge help to the people going on dialysis, but also dialysis centers who are often overrun with people who need dialysis.
Very much so. Have we made it to the end of the show?
We have.
Yeah, I think we did it. So many good stories this week. So much interesting conversation. We could have kept talking about a lot of things, trying to keep us on time, everybody. But I do want to thank you for listening to the show. Hope you enjoyed it. Also, it's time for some shout outs. Shout out to Fada. Thank you for your help with social media and show descriptions. Thank you to Identity4 for recording the show. Thank you to Gord and R and Lore and so many other moderators who keep our chat rooms happy and clean and just a wonderful community place to talk about the show and science. And I would like to thank Rachel for editing the show. Thank you for your assistance. And finally, thank you to our Patreon sponsors. Thank you to Kent Northcote, Rick Loveman, Pierre Velazar, Ralphie Figueroa, John Ratnaswamy, Carl Kornfeld, Karen Tazi, Woody MS, Andre Bassett, Chris Wozniak, Dave Bunn, Vegard Sheftad, Hal Snyder, Jonathan Stiles, a.k.a. Don Stylo, John Lee, Ali Coffin, Maddie Perrin, Gaurav Sharma, Shubru, Don Mundis, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fredis104, Sky Luke, Paul Ronovich, Kevin Reardon, Noodles, Jack, Brian Carrington, Matt Bass, Sean and Lena Lamb, John McKee, Greg Riley, Mark Hessenflow, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard, Brendan Minish, Johnny Gridley, Kevin Railsback, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Hey Arizona, Support Aaron Lieberman for Governor, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Mallory Sutter, Philip Shane, Kurt Larson, Mountain Sloth, Jim Drapeau, Sarah Chavez, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Disney, David Simmerly, Patrick Pecoraro, Tony Steele, Ulysses Adkins, Brian Condren, Jason Roberts. Thank you for all of your support on Patreon. And if you are interested in supporting us on Patreon, head over to twist.org and click on the Patreon link. And on next week's show, we are going to be joined by Dr. Michael J.L. Johnson, who will be talking with us about using metals as antibacterial, antibiotic agents.
Fun.
Yeah,
so we'll be back then talking to him, 8 p.m. Pacific time, broadcasting live from our YouTube and Facebook channels and from twist.org/live.
Hey, do you want to listen to us as a podcast? Perhaps while you swim a channel. That would be fun. Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you've heard here today, show notes, links to the stories are all available on our website, www.TWIS.org. And while you're there, you can sign up for a newsletter.
You can also contact us directly. Email Kirsten at kirsten@thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put TWIS, TWIS, in the subject line, or your email will be shouted in such a high pitch that none of us will hear it because of all of the concerts we went to as youths. Sorry.
Yes, and if you're recovering from having gotten your vaccine shot in the wrong bodily limb, you might as well reach out to us and tell us about it on Twitter, where we are at Twist Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes due in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.